Thank you, everyone. I'm sorry about my trip. It reminds me about my first visit to this meeting. It was in 2016. I came to Oslo, and as Dan said, Dada2 Foundation may not be so rich at that time, because when I should go further to Washington, I was not admitted because the credit card for Dada2.org did not work. I had to stay one night, and luckily I ordered the ticket the day before the meeting, so I made it anyway. The only thing I thought about this time was I have to go to this meeting, otherwise I would die, so I get there. So the first here is actually from my first talk in 2016, and it's the peaks of Tromsø area, the Lyngsalpene. And I think I'm going to talk about the function of ADA2, and I just think that we are to get an overview to understand the disease, we need to know the function of the protein. Without actually knowing the function of the protein, I think we are not on the top. We don't understand this disease, and we have problems also to find the therapy for the disease. So this is also from my first talk in 2016, where I said that ADA2 is a lysosomal hydrolase that transforms adenosine to inosine and may bind to DNA, RNA. And that's 7 years ago, and I think I tried to convince the Lysosomal Society in Barcelona recently for a 15-minute talk that ADA2 is actually a lysosomal protein, and it is difficult actually for such a short talk. But I think that these 7 years and all the experiments has been done since then clearly shows that adenosine ADA2 is really a lysosomal hydrolase. And at that time I suggested that the protein binds to DNA because of the heparin binding affinity, and that heparin is kind of a similar to DNA, and that's kind of a theoretical approach actually at that time. I also said at that time that the innate recognition triggers secretion of mannose-6-phosphate dependent lysosomal enzymes. I think this is a problem in a way that, if you have an inflammation, like you have a tuberculosis or a chronic inflammation, you have an increase in ADA2 in the serum. And that's actually typical for lysosomal enzymes. And it is typical for macrophages that secrete lysosomal proteins when they are activated. And I showed this last time, in 2016, that I think that macrophages secrete lysosomal enzymes when they are activated, and that can start the digestion already outside the macrophages. And that may be important as an anti-inflammatory thing, because this is a very important to get a quick degradation, because many of these components that are produced are pro-inflammatory. And also the pH outside the macrophages are acid in an inflammatory region, so these lysosomal proteins will be active. I also showed that time also that ADA2 contains mannose-6-phosphate groups, and that is probably transported to lysosomes via the mannose-6-phosphate receptor. So all in all, this is a very typical lysosomal protein, and when I purify the protein, it's an extremely stable protein, and that's also typical for lysosomal proteins. So it is really a lysosomal protein. To convince you, maybe I have to use 3-4 hours talking. I can't do it just now. The development of a therapy, of course, we have a certain therapy, but it's not, as people said, it's not curing the disease. So what we are trying all the time is to use the basic research, try to find more information, and use this to get even better treatment of the patient. And I think that's the phase we are in now. So that time in 2016, what's the next move? It's a chess game. And next move is, of course, to try to link the function of ADA2 in the lysosomes and the DNA binding with the clinical symptoms, and then to try to find new ways to treat the patients. Yeah, thanks. Oh, no, it was too quick. So I want to take your focus on cross-presentation or in macrophages. It's a kind of a mechanism where actually an exogenous protein, an antigen, when it's taken up by certain macrophages, they can actually, these peptides can actually penetrate the lysosomal membrane and get into the cytosol and be cross-presented together with MHC class 1, which usually only presents intracellular proteins. So I think this shows clearly that it's a proof of concept that antigens or proteins, macromolecules that are coming into the lysosomes and the phagolysosomes can actually penetrate the membrane, and that it may be even a kind of kind of maybe a function also that they actually have a certain leakage into the cytosol, and it can have some kind of a function. But it also means that DNA fragments and also other small molecular weight fragments can also cross the lysosomal membrane. So in the cytosol, you have the cGAS, which is a DNA sensor, and upon binding to DNA, it produces a secondary messenger, cGAMP, that binds to STING, and you get an interferon 1 response, which goes through IRF3 that was mentioned also previously. And we also have a protein CHEX1 that can degrade DNA within the cytosol and then work in an anti-inflammatory way. If you have a mutation in CHEX1 so it cannot function anymore, then you get autoinflammatory disease. And it's also recently determined also that CHEX1 actually have also a binding affinity to a protein called oligosaccharyl transferase 1, which attaches N-glycans to glycoproteins. And if it doesn't bind to OST, if you have mutation C-terminally in CHEX1, then you get an accumulation or an increase in these glycan structures that can also cause autoinflammation. So we have some kind of in the cytosol, we have a lot of sensors that can sense changes in pro-inflammatory metabolites and cause inflammation if you get an increase that is above a certain threshold. I also want to mention the work by a Japanese group, Kawana et al., who studied mice that lacked DNase2, lysosomal DNase2. They accumulate DNA in the lysosomes. And these mice get typical type 1 interferon production, especially in macrophages that are close to involved in the hematopoiesis or to the production of red blood cells. And these cells, their only explanation, because they try to remove the toll-like receptors, all the toll-like receptors, to try to see if toll-like receptors was involved in this process, but they were not. And their only explanation by ruling out all other possibilities is that DNA can, in some macrophages that are especially stressed with DNA, the DNA fragments can leak out into the cytosol, bind to C-gas, and you get the type 1 interferon response. And importantly here is that these mice can be treated with anti-TNF-alpha treatment, and they get much better. So it may be actually a similarity with DADA2. So I also want to mention the project I was working with previously, lysosomal alpha monosidase and alpha monosidosis. That disease is resulting from a defect in the degradation of N-glycans, and you get a tetrasaccharide in cattle and cows and cats, I mean. Whereas in human and mouse, you get the trisaccharide. And the clinical phenotype is very different between these two. They have very much stronger phenotype, where you have involvement of the CNS in cattle and also in cats. And it is a kind of a possibility that it is actually the tetrasaccharide may have more pro-inflammatory function than the trisaccharide that you find in humans and mouse. So it might be actually a link between lysosomal storage disorder and DADA2. So what I want to conclude here is that what if a small molecule in lysosome can cross the membrane and penetrate into the cytosol and bind to cytosolic sensors? This would mean that we have actually an array of lysosomal proteins that play a very important role in the regulation of inflammation. They regulate they downregulate inflammation by degrading, and they prevent the accumulation of pro-inflammatory molecules in the lysosomes. ADA2 is a DNA binding protein, and by binding to DNA, it can also prevent free DNA to leak into the cytosol. So it is a possibility. And if this is correct, it would mean, I think, a new era of understanding inflammation, where you put actually DADA2 into the concept of lysosomal storage disorder, and you can have a bigger understanding of what is causing the clinical phenotypes of the lysosomal storage disorders as a whole. Yeah, that's where the point is. Yeah, there we are. So just very quickly, the last part of my talk. ADA2 is very conserved in eukaryotic, among eukaryotes, except maybe in plants. Yeah, point, yeah. And yes, the isolated point is very high, it's around 9 in mammals. So it means, I think it conforms to a role of ADA2 in the binding of DNA. And I think it's changing through evolution, so it's only in mammals you have this very high isolated point. So it comes to a point here where you, yes, thanks, where you try to find that things may be happening during evolution, that in the mammalian evolution, the activity, adenosine deaminase activity decreased or changed dramatically. And it indicates maybe that ADA2 has actually gained a new function in the mammals that you don't find in the earlier part of the evolution. So we cannot maybe not say what is the function of ADA2 in general, but you must look at what is the function of human ADA2, and that may be different than the function of, for example, ADA2 from the insect. So we have to change, and that's why I try to make my lab like a zoo, So I tried to investigate the ADA2 from many different mammalian species. Yes. This is. So I think I will conclude then by this last maybe provocative slide here, where I think that the evolution of ADA2, you must look at it by the possibility that it was actually first during early evolution, it was an extracellular adenosine deaminase. Maybe later it got also a slight DNA binding activity, maybe by some positive charges, and then maybe that the protein developed. But when the adenosine deaminase activity was not important anymore, you get some kind of a certain small mutations that kind of decreases the activity of the protein, of the adenosine deaminase activity, and maybe you can get some what I call a phantasma ADA2, that you have a protein that actually don't have any function, and later in evolution you get a strong DNA binding, and then it turns out that the body adapts so that it gets an important function in the DNA binding and then regulation of DNA to prevent DNA from escaping into the cytosol. So that is the major conclusion. So I will not interrupt you with the lunch, and I think we have a nice discussion later.